Good morning and welcome to all B.Sc. first year students of Fishery Science in online lecture series of semester 2nd, paper 4th, Fish Seed Production Technology and Hatcheries Management. In this paper, we have seen different methods for the seed production. As required demand of seed production or the essential demands for the fish seed is to be day by day, these are to be increases, but seed availability, egg availability, these are to be less. To fulfill these required or the requirement of egg and seed, these fish seed or the fish egg, these are to produce on a large scale. Different methods which is to be you have select different method which is to be you have seen, in which natural method, second bund breeding, different types of bunds, and then artificial fertilization by stripping method, and last method, most important method, that is the induced breeding by hypophysation. In this induced breeding by hypophysation, first we have see the definitions of induced breeding, and then we have see that is the collection of brooders, where brooders these are to be collected. In last part, brooders, the mature fish, these are to be collected either from the wild source, mature fish these are to be collected from the natural way in those ponds, lake, rivers, reservoirs. Fish these are to be available from these sources. Fishes these are to be collected, these are to be captured. Second, fishes these are to be captured from the fish farming or the fish farming areas, and these fishes these are to be collected either from natural sources, fish farm, or these fishes these are to be collected from the wild sources. Then these fishes, care should be taken that which are the care should be occur and which are the care should be taken. These care is to be should be taken. In this part, we have see that brood fishes these are to be collected, brood fishes these are to be selected. Then what is the care for these mature fishes? Care should be taken. Care should be taken, and that's why care of brooders is essential. Care of brooder is to be most important, which is to be we have seen today's part. Care of brooders. The brood stock pond should be prepared. The brood stock pond should be prepared following standard procedure to ensure sustained production of zooplankton. The brood pond or the brood stock pond should be prepared following standard procedure to ensure sustained production of zooplankton. Stocking rates are manipulated. Stocking rates are manipulated to permit individual and collective care. To permit individual and collective care of brood fish. Those are to be ready for spawning. Those are to be ready for breeding. Brood fish, enabling them to get nutritional and environmental. Enabling them to get nutritional and environmental advantages for onset of right degree of maturity. For onset of right degree of maturity means enabling them to get nutritional and environmental advantages for onset of right degree of maturity. During immature stage, feed the fish with a traditional diet consisting of rice bran and oil cakes. When these immature brood stock is to be collected, immature brood stock is to be captured. These immature brood stock that can be fed with a traditional diet, that is a traditional diet. That traditional diet consisting of rice bran and oil cake. These rice bran and oil cake in a ratio of 1:1. 1 part rice bran, 1 part oil cake, and this 1:1 ratio of rice bran and oil cake they can be fed to these brooders at the feeding rate of, what is the feeding rate of these brooders? The feeding rate of these brooders is to be 1:2%, 1 to 2% of its body weight daily. Those mature fishes, these are to be immature fishes, these are to be select, these are to be choose. These immature fish provide a diet, traditional diet, and this traditional diet, these are to be consist of rice bran and oil cake. Rice bran 1 part, oil cake 1 part, and 1:1 ratio of this supplementary food, artificial food, these are to be supplied, these are to be provided to these immature fish, and feeding rate is to be 1:2% of its body weight daily. 1 ते 2% त्यांच्या वजनाच्या डेली आपल्याला काय करायचं आहे, आर्टिफिशियल फूड किंवा सप्लिमेंटरी फूड, दीज आर टू प्रोव्हाइड. ड्युरिंग द मॅच्युरिंग फेज, feed these fish. During the maturing fish phase, feed the fish with a special feed containing rice bran, oil cake, fish meal, cereals, grams, and mineral and vitamin mixture. That is, when fish is to be maturing phase, at that time the food grain, these are to be supplied, and these food grain, these are to be a diet. That diet is the nutritional diet, that diet is the balanced diet. What is in the balanced diet? In this diet containing rice bran, oil cake, fish meal, cereals, grams, minerals, and vitamin mixture. Means all these are to be mixed in diet, and such diet these are to be provided, such diet these are to be supplied to the brooders in maturing phase. When such balanced diet these are to be supplied, fish can be grow within short period, within short time. Fishes, they can grow, they can achieve maturity, they can achieve a growth. Alternatively, one can use commercial floating pellets feed. Alternatively, we can use commercial floating pelleted feed is to be provided, and that commercial floating pelleted feed, it is to be contain protein content near about 30%. And when such alternatively commercial floating pelleted feed, these are to be contain, which feed these are to be provided, which contain 30% protein, fish can mature early, and fish can attain a maturity within short time, within short period. Yes, within short time, within short period. In addition to artificial feed, grass carp is also given. Tender aquatic weeds, grass carp is also given tender aquatic weeds per terrestrial grass, and these tender aquatic weeds per terrestrial grass, these are to be provided, these are to be supplied to these grass carp or to these fishes. Breeding habit of some species like common carp, breeding habits of some species like common carp demand their separation from other carp species. Due to their natural breeding in ponds with aquatic vegetation, breeding habits of some species like common carp demand their separation from other carp species due to their natural breeding in ponds with aquatic vegetation. Because these exotic species, they can, or common carp, it can breed in aquatic vegetation, where that is aquatic weeds, these are to be more in water. In such water, these fishes, these are to be breed, these are to be spawned. As a result, the common carp brood fish, is segregated sex-wise and stocked in separate ponds to prevent accidental spawning in ponds. As a result, the common carp brood fish is segregated sex-wise and stocked in separate ponds to prevent accidental spawning in ponds. However, the rest of the species can be stocked in a commercial pond or stocked in separate ponds after species wise and sex wise, or it is to be a segregation needs to be done. That segregation is to be either by species wise, either by sex wise, depending upon the conditions of our culture, conditions of our rearing and breeding. These captured mature fishes that can be stocked communal ponds in or stocked ponds, separate ponds, depending upon their species wise or sex wise. Cutla in particular needs to be separated from the rest of the species as it shows poor response to hormonal injection when stocked with other species. When katla, these are to be stocked with rohu. When katla, these are to be stocked with mirgal. Then these fish, these are to be do not show a hormonal injection or do not give response to the hormonal injection in these communal species. That's why when katla, these are to be cultured, these are to be separated in the rest of species. It shows that poor response to hormonal injection when stocked with other species. That's why katla is to be kept in separate ponds, separate tanks. A gravid fish when held by hand, with tail up should particularly ooze milk and also oha. When we have observed, when we have seen that is fish is to be mature, ready for oozing out, ready for spawning, at that time these fish, these are to be taken in hand in such a way that tail is to be in your hand and fish is to be, its head is to be on lower side. When such condition, these are to be seen. A gravid fish when held by hand with tail up should practically ooze milk and also oha. It is the sign, it is the symptom for oozing out for the mature condition of that is fish. The paddle wheel aerator, particularly in katla pond, can provide additional aeration, particularly during morning hours. During morning hours, when katla, these are to be done, when katla, these are to be performed a breeding season, in this paddle wheel aerator is to be added, and with the paddle wheel aerator, particularly katla pond can provide additional aeration, because for katla more oxygen, these are to be essential, more oxygen, these are to be required during the monsoon period, and due to this paddle wheel aerator, more oxygen, these are to produce in those pond in which katla is to be stored, Segregation of sexes at least 1 month before increases the affinity between male and female during spawning. It is again interesting. When male and female, these are to be segregated, when before 1 month of spawning, before 1 month of breeding, both male and female sexes, both male and female fish, these are to be segregated at least 1 month before the spawning. When these are to be before 1 month, these are to be segregated, then affinity of these male and female, these are to be increased, and when affinity, these are to be occurring male and female fish during spawning, they can meet intimately and schooling behavior as sexual play, these are to be performed, and when sexual play, these are to be performed, spawning, these are to be done carefully, spawning, these are to be done fully. Care should be taken to maintain water quality. Care should be taken to maintain water quality and plankton level by periodic manuring. Care should be taken to maintain water quality and plankton level by periodic manuring, that is at 1/10 of initial dose. 1/10 of initial dose. These maintain water quality and plankton. To maintain phytoplankton as well as zooplankton, these are to be maintaining in water. Algal blooms and oxygen depletion are controlled by water exchange. Algal blooms and oxygen in those water in which these fishes, these are to be kept. In these water, algal blooms, these are to be not grow, as well as oxygen level is to be not depleted, means to minimize algal blooms and to increase the oxygen level, that water can be exchanged regularly, water can be controlled, exchange activity of water regular manner, regular way. Parasite and pathogens should be controlled. Parasite and pathogens should be controlled by periodic checking of brooders. That is, care should be taken. Parasite and pathogens should be controlled by periodic checking of brooders. Those brooders, these are to be collected. Those brooders, these are to be selected. These brooders that can be continuously controlled, that can be continuously checking by these brooders. No any parasite, no any pathogen, these are to be attacked. Suppose parasite, these are to be affected, these are to be attack on the body, that parasite can be removed as possible as early. Or those pathogens, these are to be affected on the fish, these pathogens can be removed out with chemical method, biological method, or with physical method. Means when pathogens, these are to be removed, that can be safe, that can be good for these brooders. The common parasites like Lernia and Argulus are the common parasite like Lernia, Argulus are common on major carps. Cutla is more susceptible to Lernia and Arbulas, because these Lernia and Arbulas, that is, these are the crustacean parasite. These crustacean parasite, these are to be more common in carps, especially in Cutla, and Cutla is to be suffer, Cutla is to be attacked with these Lernia and Arbulas, and can be controlled by manually, as well as it can be controlled by removing the disinfecting the affected fish with a solution of KMNO4 solution about 5 pm. Means those pathogens that may be bacterial pathogen, that may be fungal pathogen, that may be helminth pathogen, or that may be, that is, worm pathogen. These pathogens means those fishes, these are to be select for breeding. Those fishes, these are to be choose for breeding. These fishes, these are to be free from diseases. These are resistance powered, these are resistance fishes, and these are to be free from pathogens, free from parasite. Then and then they can spawn, they can breed a good quality and quantity of egg without attacking parasite. Means these are the different care should be taken. These are the different care, that may be parasite, that may be algal blooms, that may be oxygen level is to be to maintain, water level is to be maintained, manuring is to be done continuously, then separation of male and female, then before 1 month male and female, these are to be keep into the pond, affinity is to be increased, then aeration for the oxygen, aeration is to be provided, aeration is to be provided, then in addition artificial food, these are to be supplied, then breeding fishes, these are to be keep into the some aquatic vegetation. As a result, sex wise, species wise, these are to be occur. These separate prevent accidental spawning. Means such different care should be taken. When fish, these are to be keep, when fishes, these are to be stocked in breeding pond. In breeding ponds, tanks, a care is to be maintained, a care is to be essential. Without maintaining care, without taking care, these brooders, these are to be not fully, forcefully or healthy spawn, and when these are to be not spawn, then our aim of egg production is to be fall down, and when our aim of egg production is to be fall down, required quality and desired quality of seed is to be not produced. That's why to maintain or to produce essential requirements, or to produce the required quantity and desired quality of fish seed, care of brooders is essential, and with this above methods, care should be taken, care should be done by fish farmers. With this, we have completed this article, that is care of brooders. Then in next part, we have see the next article, how these brooders, these are to be identified, identification of brooders, male fish and female fish, how we can identify, we can see in the next part. Hope you have understood up to here. Any problem, please ask. Thank you all of you.